Okay, so we're going to hear from Max Khachikian.
Yeah.
Aliya Zhou, Max Steiman and Ethan Trinh.
Yeah.
Exploring the role of NAFTA and nitrogenase regulation. Presented by Aliya Zhou.
I'm Max Khachikian.
Ethan Trinh.
And Max Steiman.
So we worked under Dr. Beccah Warmack in the Rees lab. And before getting into our research, I'd like to give you a general background. What is nitrogenase? Nitrogenase is a metalloenzyme, meaning it is an enzyme comprised of metal ions, and its substrate is dinitrogen. It takes dinitrogen and converts it into ammonia through a process called nitrogen fixation. Nitrogenase's structure has two main components. Component 1 is iron, and component 2 is molybdenum iron protein, or the MOFI protein. And these work together in nitrogen fixation, and the iron component gives the MOFI protein the needed electrons to complete the process of nitrogen fixation. Nitrogenase is found in a handful of prokaryotes, and prokaryotes are bacteria and archaea.
Why do we care about nitrogenase? So the Haber-Bosch process, shown on the right, is the leading industrial process for the production of ammonia. And ammonia is commonly used in plastic production, fabric production, and even pesticides. Now, this process, while it is very helpful, is also very detrimental because it produces a lot of pollution. 2% of all global carbon emissions come from that process. So if there's a possibility to have nitrogenase be mass produced to completely eliminate this process, that would be very beneficial to the environment. Nitrogenase is also a very large factor in the nitrogen cycle.
So nitrogenase has to be worked with in an anaerobic environment, which basically, because contact with oxygen deactivates the nitrogenase, which means that we get to work in these super cool tents that are filled with argon and are basically completely free of oxygen.
Right, so we identified a protein called NAFTA using structural proteomics, where we study structures of different types of proteins in a bacteria called Azotobacter vinyllandia. So basically, short story, we first purified the nitrogenase MOFI protein from a mutant Azotobacter strain that lacked the NIF that was responsible for containing and producing the homocitrate synthase. So without this gene, the MOFI protein cannot insert homocitrate into the active site and perform its function. Now, we ended up calling this protein the MOFI-NIF-B. During our structural proteomics analysis of MOFI-NIF-B, we discovered an additional density in the structure, which is this green part right here. And this uncharacterized protein is known as the nitrogenase associated factor T, or for short, NAFT. And we're curious of what this protein does. That was like our research, basically. So how do we identify this MOFI-NIF-B protein? So taking a closer look at this protruding density from the MOFI-NIF-B protein in green, Beccah basically built a model of the green density structure and then compared it with the mass spectrometry results, just the text in the blue box, and was able to positively identify the green density as the nitrogenase associated factor T. So we studied the endogenous NAFT complex structure in more detail. So if you look at panel A, that's basically the same thing, or another viewpoint of the protein NAFT complex from the previous slide. And panel B is basically looking at the fit of the model, which is shown in green inside like the little map, the gray mesh. And this panel shows that NAFT is a good fit with the density, which is further evidence that we have correctly identified the green blob as NAFT. Panel C is the overlay of the NAFT and MOFI complex and the MOFI iron stabilized structure. The green blob partially overlaps with the MOFI iron stabilized structure, shown in gray, which indicates that these two proteins cannot bind together at the same time. With this information, we can also imply that the NAFT and MOFI protein are not active. In panel D, if we look at the interface between the MOFI and NAFT proteins, we see an interesting cluster of histidines, like the lines, and this basically indicates a metal binding site. Furthermore, there is the female cofactor nearby, so it's possible that NAFT is potentially trying to grab onto the female cofactor.
So our presentation is going to have two parts, and the first part is isolating purified NAFT for experimental characterization. So in order to further analyze NAFT, we need a lot of NAFT, and how we did this was a plasmid that coded for NAFT. We transformed it into E. coli, and we did this by heat shocking the E. coli to make the membrane permeable, and then we exposed it to NAFT, so the E. coli now has NAFT inside of it. We then ran some gels under various conditions to see which condition had the best production of NAFT. Our conditions were 37 Celsius and 17 Celsius, either at 0.2 millimolar IPTG or at 1 millimolar IPTG.
All right, and here's the specific process of that entire experiment. So once we have the E. coli cells containing the NAFT tags, we let the NAFT express in the E. coli cells, which really just means we kind of let the E. coli cells grow and develop. And then we took samples of the different E. coli cells under different conditions. We put them in tubes, and then we centrifuged the tubes. And what that does is it separates the heavier cells, and those end up as pellets at the bottom of the tube. And then we added blue dye to the pellet and resuspended the pellet. And finally, we added that solution to an SDS gel. And SDS page gel is a chromatography that separates all the proteins in a cell by weight. And NAFT is like right there, the 15 one. And the result is the band corresponding to NAFT appears to be the strongest in the 37 degrees Celsius, 0.2 millimolar IPTG condition.
So now that we've identified the condition that it expresses at best, we are going to try to isolate and purify the NAFT protein. And we do this by using the same process in the previous slides to create 4 liters of NAFT expressed in E. coli through adding IPTG. We then take these cells and lyse them, which is essentially breaking them apart into their subunits. And then we spin this down to create a pellet and a supernatant. We don't care about the pellet, and the supernatant has the NAFT inside of it that we want. So we send the supernatant with the NAFT in it to a company outside the lab, and they add a strep tag to the NAFT. And then we take our supernatant with the strep tag NAFT and run it through a column. And through this process, the strep tag binds to the column, and everything else we don't care about becomes flow through, which is discarded of. And then we put our column through a series of buffers. The first buffer, buffer A, is a wash buffer, and it discards of all the protein that are unbound to the column but somehow stayed behind, and that becomes flow through that we discard of. And then we use a second buffer, elution buffer, which has a high concentration of biotin, and biotin competes with the strep tag for space on the column and eventually pushes off the strep tag NAFT, so we are left with our isolated NAFT. Then we use a gel to test if this was successful by putting it through a gel. And if you look at the 15 kilodalton marker, we know we're successful if there are blobs there, because that's where NAFT appears. And looking at the gel, there are no blobs near the 15 kilodalton marker. So this was a failure, the first test. And this is because our protein aggregated, so there was no NAFT in the soluble fractions, which basically means there was no NAFT loaded into the gel.
Figure out why this further happened, we ran some solubility tests with NAFT. So we did the same process again, but then we used 2 different conditions. We used our 37 Celsius 0.2 millimolar IPTG, but we also ran a 17 Celsius 0.2 millimolar IPTG. And we found that in the 37 Celsius, NAFT was not soluble at all and was just not showing up on the gels. But it was soluble in the 17 Celsius and it was showing up on the gels. So then we proceeded our experiments using the 17 Celsius strand.
So using these new conditions, we tried a second purification test where we used the exact same steps in the first test, except added salt to the buffers, because adding salt to the buffers is supposed to increase the success rate of strep tag purification. And if you look at the gel, This time it was successful. There are the blobs at the 15 kilodalton marker, which means we were able to successfully isolate and purify the NAFT protein.
So now you may wonder, why do all these steps to like characterize NAFT matter? Well, NAFT is associated with the damage state of the MOFI protein. So it could be related to some sort of regulation of the MOFI protein or recycling the female cofactor or keeping the MOFI protein stable while it is damaged and wait for it to somehow be fixed by other proteins. But there are a lot of possibilities that for the NAFT protein. However, we can't really study the protein unless we have it isolated. So all of our experimentation results basically optimize the protocol to express and purify NAFT for the future. And now moving on to the second part, exploring effects of cellular stress conditions on Azotobacter vinelandii, which I will probably just refer to as AV from now on because like try saying Azotobacter vinelandii like 5 times, it does not work. So why are we testing cellular stress conditions? Well, when we found NAFT, it was under the AV cells were under stress conditions since the NIFV gene was removed from the cell. So we wanted to see how does like different stress conditions somehow impact nitrogenase. So we have the list of conditions right there. There's low temperature, high temperature, low pH, high pH, and low citrate. And obviously there is a control. So once again, we let the NAFT express in the AV cells, and then we put the AV cell samples in a vial with a rubber stopper. And then we introduce acetylene into the headspace of the vials. Acetylene is a substrate for nitrogenase. And nitrogenase converts acetylene to ethylene. So by observing how much ethylene there is in the vials, we can observe how much activity the nitrogenase is kind of doing. So conclusion, most of the conditions resulted in a decrease in nitrogenase activity. However, high pH and lower concentrations of citrate appear to have like somewhat the same amount of activity as the control.
So there are various ways to study these stress conditions, and one of them is through morphology. Morphology is looking at how stress conditions can change the size and shape of the cells. So the image to your right is of mutated Azotobacter vinelandii, and it is mutated by adding a UNA-G tag to it. And UNA-G was initially found in eels, and when bilirubin is added to UNA-G, it fluoresces, which is shown in the image in the center. So what we did is we added the UNA-G tag to nitrogenase and took a fluorescent image on a fluorescent microscope. And this is interesting because it shows not only how the stress conditions can affect the size and the shape of the cell, but also how it affects the location of nitrogenase in the cell. And sadly, because of the short amount of time we had together, we weren't able to complete this process of studying the various stress conditions, but we think it's important and should be studied in the future.
So our conclusion. So we used a combination of molecular biology, biochemical techniques, and including bacterial culture, transformation, gel electrophoresis, protein purification, gas chromatography, and the fluorescent microscopy to optimize the purification of the recombinantly expressed NAFT protein and to identify and characterize the initial cellular growth conditions under which Azotobacter vinelandii experiences stress. These are acknowledgements. Thank you to the Rees Lab, Doug Rees, who's sitting next to my younger brother, and Dr. Warmack, who is not here. And questions?
Questions?
So what was your biggest surprise in all?
I'd say the biggest surprise was when we ran the purification initially and it didn't work out because we were really confused. We didn't know why, and we ran it like twice or 3 times, and each time it wasn't working. So that was the biggest surprise.
Can you guys be reasonably sure that the NAFT by itself has the same, like it has the same conformation structure, like is folded the same as it is when it's associated with nitrogenase? Yeah.
It is highly unknown right now, and like, because this is kind of, it's just like unknown territory, so we actually have no idea.
Most of what we did is just like how to isolate the NAFT.
Mhm.
We haven't really gotten to the like investigating what it actually does.
All right, future studies. Mhm.
I was impressed by the choreography of.
Yeah.
That was